finally, some much needed alone time. Hello, pleased to meet you. 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 Hello, hello.
Tens of millions of microorganisms have been discovered throughout the years, thanks to him and his small but terribly amazing microscope. Microorganisms' motility are deeply observed, particularly microbial dispersal. We all know that microbial dispersal in an aqueous environment can be active or passive, based on the mechanism that drives the dispersion. In active dispersal, individual cells are equipped with motile organelles such as flagella or pili, which enable swimming in liquids or gliding motilities near a surface, respectively. While in passive dispersal, the dispersal units consist of a sessile cells alone, which lack self-powering motile organelles and can only be transported through environmental entrainment. Do you ever wonder if sessile cells can have the ability to move? Well, somehow, or do they actually move without us knowing? Well, there is a potential for a sessile-motile coexistence in rosettes that is primarily driven by the asymmetric cell division, where each newly yielded daughter cell grows a functional motor and a flagellum. In a speculation where these rosettes, the aggregates of sessile stalk cells, are formed by random collisions between stalk cells at similar stages of growth, which attach to a common core through holdfast. So which bacteria has an asymmetric cell division and uses rosettes to study its mechanical properties and mechanisms of adhesion? The researchers come up with Caulobacter crescentus because among Caulobacter species, the non-pathogenic strain Caulobacter crescentus has been most studied primarily for understanding its dimorphic life cycle and its asymmetric division. The life cycle of a C. crescentus begins as a curved rod-shaped motile cell known as a swarmer. This motile stage eventually ends with the swarmer cell shedding its flagellum and growing a stalk. The cell then elongates its body and becomes a predivisional cell, which later undergoes asymmetric division and gives rise to a stalk mother cell and a flagellated daughter cell. A motile cell is propelled by the rotation of a single helical flagellum, which is powered by a flagellar motor that can operate in both clockwise and counterclockwise directions. After a finite division time, typically over an hour, is expected to cause intermittent bimodular states of rosettes that consist of both sessile and motile members, and that collective motility can arise from the aggregation between sessile and motile compartments. But this is just a hypothesis. To confirm this, the researchers using and Binliu come up with their study titled Self-Propelling Enrolling of a Sessile Motile Aggregate of the Bacterium Caulobacter crescentus. Methodology: Swarmer cell and rosette preparation. Wild type of Caulobacter crescentus were used, cultured in peptone-yeast growth media at 30 degrees Celsius. Single colony from an agar plate was inoculated and incubated before being transferred to a petri dish and incubated overnight. Swarmer cells were obtained using a plate releasing technique. Two methods were used to obtain rosettes in this study. First, the researchers added 2 microliter of the medium containing swarmer cells into the growth medium in a flask, which was then incubated within a shaker with 50 revolutions per minute at 30 degrees Celsius for 18 to 20 hours. Second, the researchers prepared Caulobacter crescentus culture on an agar plate and transferred 0.1 microliter of the culture into 100 ml growth medium, which was then incubated within a shaker with 50 revolutions per minute at 30 degrees Celsius for 18 to 20 hours. Rosettes of typically 5 to 10 micrometer in diameter were generated under both culturing conditions. Second, experimental filming. The culture was diluted by 2 to 3-fold and a drop of the sample was sealed between 2 cover slips using vacuum grease. Swarmer cell and rosette imaging. Filming was conducted using an inverted phase contrast microscope. For Caulobacter crescentus swarmers, the microscope stages were programmed to move along with an individual cell in 3D to maximize the tracking time. For Caulobacter crescentus rosettes, the relatively slow movements were followed by manually adjusting the microscope stage in 3D to avoid mistracking due to variant appearances. Fourth, scanning electron microscope. Culture medium with Caulobacter crescentus rosettes was first fixed using 3% glutaraldehyde in 0.1 M phosphate buffer for 1 hour. The sample was then filtered using polycarbonate filter. After 3 washes in buffer, the sample was dehydrated in an ethanol gradient. The sample was then critical point dried and spatter coated with gold. Samples were then mounted on SAM stubs and examined with a ZEISS Gemini 500, given partially damaged flagella in preparation, and the inaccessibility of a bottom view. This protocol was not used for counting the total number of flagella, but for confirming the presence of flagella in cultured rosettes. For rosette morphology, to measure rosette size, the researchers recorded Z-stack images of each rosette by scanning the sample along the Z-axis using the piezo stage. Swarmer cells and rosette dispersal. The motility of a rosette near a solid surface and that of a swarmer were both characterized by their 2D mean square displacement or MSD in the XY plane. In the swarmer cell case, due to the relatively thin sample thickness and the relatively high swimming speed, the actual movements were geometrically restricted to be quasi-2D. To avoid the potential bias from short rosette tracks, rosettes that stayed active within the view for over a minute were used for MSD calculations. Rosette kinematics. Rosettes were treated as rigid bodies due to the high stiffness of stalks and hold-fast adhesion. The 3D trajectories of rosettes were reconstructed by integrating the offset of the rosette from the image center, instantaneous stage position, and the estimated focal depth of the microscope. And lastly, statistics and reproducibility. Experimental data in this study were collected through multiple independent trials, and their values were characterized by mean standard deviations provided together with the corresponding sample sizes. Statistical tests were performed by standard regression, linear and nonlinear algorithms coded in MATLAB, and pairwise comparison using one-way analysis of variance or ANOVA coded in R49. Results and discussion. As shown in the typical rosette image, sessile member cells undergoing different growth stages appended to each other through their stalks near the center of the rosette. Here, a single prodivisional cell was found equipped with a flagellal filament representing the motile compartment in the sessile motile aggregate. The restoring time characterizes the reorientation timescales of both solitary cells and rosettes, as indicated by the much of the crossover times within the ballistic and diffusive regions revealed by the MSD analysis. The similarity in motor behaviors further indicates that rosettes can be each powered by a single flagellated member cell. The restoring time characterizes the reorientation timescales of both solitary cells and rosettes, as indicated by the much of the crossover times within the ballistic and diffusive regions revealed by the MSD analysis. The similarity in motor behaviors further indicates that rosettes can be each powered by a single flagellated member cell. The typical rosette trace resembled a random walk but consisted mostly of curved paths rather than the linear segments found in solitary swarmer cells. In addition, rosettes move at a ballistic speed much slower than that of individual swarmer cells. Figure 3 shows that even for a fixed angle between these two vectors, the projection of this angle onto the XY plane is arbitrary for a rosette swimming in the bulk, subjected to the orientation of the plane formed by these two vectors. Figure 4 shows when a motor switches direction, the rosette reverses its direction of translation and almost retraces its previous path. This forward-backward reorientation is distinct from the run-reverse flake motion of the solitary swarmer cells. For the conclusion, collective motility can arise from the aggregation between sessile and motile compartments. The probability of having more than one active flagellum is as low as 3%. Rosettes showed higher probabilities for relatively longer motor durations. Through reconfigured powering and locomotor mechanisms, an aggregate like C. crescentus rosettes enable transport of otherwise stationary cells. Application of the study. The aggregation-based motility described here offers a spectrum of implication for understanding the potential diversities in microbial motility. For example, aggregation-based dispersal may facilitate pathogen transmission. Second, in pathogenesis, many infectious microorganisms known to disperse passively can disperse more rapidly if aggregated with motile cells, as evident by this study. Additionally, the aggregation between microorganisms and non-living particles may also exhibit active motility and enhance the transport and exchange of environmental particles. Third, following the architecture of a sessile motile aggregate presented here, an aggregation-based design with modularized motor and structural components can be integrated with the existing microrobotic systems for self-propelled and boundary-associated transport.